All right, so this is the brain chapter, the final of this third unit. We've done the nervous tissue, the spinal cord, and now we're looking at the brain. And the focus of this lecture is really going to be just sort of getting a sort of anatomical orientation on the brain in terms of the different regions and generally what they're sort of responsible for. And we'll take it sort of segment by segment after we learn about some of the sort of ways the brain is protected. So, as I've said before, the human brain obviously is much more complex than what we see at the spinal cord. Spinal cord really was more about sort of conduit of information and processing of reflexes and things like that. Obviously a very essential tissue, but not near as complex as we see at the brain. So 97% of the body's neural tissue is found in the brain, which consists of tens of billions of neurons. So all of the really higher level, higher order functioning of the brain, of neural processes is going to occur in the brain. And so the 6 regions of the brain, as I said, we're going to sort of try to get a feel for how it's organized. The 6 general regions are the cerebrum, cerebellum, Diencephalon, Mesencephalon, Pons, and then the Medulla Oblongata. And if you look at this image, you can sort of see generally how it's organized. Basically, where the spinal cord ends is where the Medulla Oblongata begins, right there. And so that's the same side as the foramen magnum at the skull, where the spinal cord begins as it exits the skull and begins the spinal cord in the C1 of the cervical vertebrae. So, what we're going to do first is just get a feel generally for what these different regions do. Cerebrum, which is on top, it's the largest part of the brain. As I said, this is where all the higher level functioning occurs. It's divided into 2 distinct hemispheres, left and right. It has a surface layer of what we call gray matter, which is also referred to as neural cortex. There are also some deeper areas of gray matter. Those are referred to collectively as the basal nuclei. And then everything else is going to be white. So I think I talked about seeing a cross-section of the nervous tissue. You're going to have gray matter at the very top, that's your neural cortex, and you're going to have segments of dark matter much deeper in, and that is the basal nuclei. Cerebellum is the second part, largest part of the brain, also referred to sort of as the mini brain, right, because it's organized similarly in that it has 2, hemispheres, and it's completely separate, but is much smaller and sits just below the occipital lobe and functions primarily in regulation and coordination and fluidity of movement. And so we'll talk about the cerebellum and how it regulates motion and movement, things like that, coming up. All right, so, and you can see those 2 components here. Cerebrum here, cerebellum here, right? So cerebrum is this big, large component. This is where it has all those higher level functions, conscious thought processes, intellectual functions, memory, emotion, cognitive ability, etc. All these things are located in the cerebrum. Cerebellum, which sits right beneath it, right, which is this guy, is more regulating and adjusting output of somatic input, motor patterns, fluidity of movement, things like that. So those are the 2 larger portions, cerebrum and cerebellum, and then as you can see, that's housed underneath that really is the brain stem, and then the diencephalon sits on top of the brain stem. So the diencephalon, as I said, links the cerebrum with the brain stem, so it's sort of the last line of communication between the 2. And is made up of 3 parts, the epithalamus, thalamus, and hypothalamus. Thalamus you can think of as sort of a relay or processing, sorting station of sensory information that's headed towards the cerebrum. Hypothalamus is a major control center, has the ability to produce hormones, and it works with the pituitary gland, which is the hypothalamus is attached to be what's called the infundibulum. And we'll see this really cool interfacing of the nervous and endocrine systems in this sort of relationship that the hypothalamus sort of has over the pituitary gland and sort of uses the pituitary gland to control a lot of regulatory processes and functions in the body. Right? So the diencephalon, as I said, sits on top of the brainstem. The brainstem is made up of the midbrain or mesencephalon, pons, and then finally the medulla oblongata. And again, where the medulla oblongata ends, then it turns into the spinal cord. So midbrain or mesencephalon, we're going to see some processing of sight and sound. Reflexes also helps with maintaining consciousness. The pons is a major connector for the cerebellum with the brainstem and also has some visceral motor control. Pons has several cranial nerves that emanate off of its structure. And then finally, the medulla oblongata, as I said, is the last part of the brain. It's what attaches to the spinal cord. It relays information from the spinal cord up or from higher up down to the spinal cord, but also via cranial nerves can regulate some autonomic functions like heart rate, blood pressure, digestion. A lot of that is done through cranial nerve number 10, the vagus nerve, which you guys will be learning coming up when you do the 4th lab unit. All right, so here's a shot of all those components, right? So here is your diencephalon, right? Sits on top of the midbrain, which is right here, pons, and then the medulla. And incidentally, you can also see all these structures are the cranial nerves that are coming off both the pons and the medulla. And this gives a nice summary of what's happening there. Obviously, we're going to go into much greater detail on all these things, but at least gives you a general idea as to what we'll be looking at. All right, so in the spinal cord, we talked about the central canal of the spinal cord, which was an opening down the middle that was lined by epidermal cells and produced CSF. Well, that same pathway, as it moves up through the medulla and up into the brain, that pathway will expand in larger chambers called ventricles. And there are actually several of these ventricles. You have what's called a lateral ventricle, which there's one of those in each cerebral hemisphere. You have a third ventricle, sort of like the lateral ventricle, kind of like 1 and 2, and then you have the third ventricle. And then you have a chamber that links down to the 4th ventricle called the cerebral aqueduct, and then the 4th ventricle, which sort of sits in between the pons and the cerebellum in terms of its location. All right, so this ventricular system, again, is just an extension of the central canal of the spinal cord that we saw when we looked at the spinal cord. Okay, and so lateral ventricle, I said there's one in each cerebral hemisphere, and it's divided by what's called the septum pellucidum, which is this thin medial partition. You'll be able to see that when you do your brain dissection. That then dumps into, via the interventricular foramen, into the 3rd ventricle. 3rd ventricle then dumps into the 4th ventricle via the cerebral aqueduct, and the 4th ventricle again extends in the medulla, and then ultimately becomes continuous with the central canal of the spinal cord. And so here are some nice images of the ventricular system. You can see it starting with the central canal of the spinal cord and continuing up into the 4th ventricle, 3rd ventricle, and then the lateral ventricles, which again, there's one in each hemisphere. Okay, and this is an anterior view of that. Again, you can see the central canal of the spinal cord and see how it expands up into this 4th ventricle, 3rd ventricle, and then you have the lateral ventricle in each hemisphere. So, sort of sum up, this brain is this very large but delicate, very delicate mass of neural tissue that needs to be protected and isolated. Contains internal passageways and chambers filled with CSF, and each of these regions has specific functions, right? As we go from the medulla and work our way up to the very top of the cerebrum, things are going to become more complex, right? It would be fairly basic down at the medulla and the pons, but by the time we get up to the diencephalon and thalamus, we're going to see much more complex functions. And so conscious thought and intelligence are all produced in the neural cortex, the surface tissue in the neural cortex in the cerebral hemispheres. All right, so now that we have a general understanding of how the brain is organized, it's also important to understand that the brain is protected and understanding how the brain is protected and supported. And so there are 2 general categories, ways in which the brain is protected and supported. You have physical protection of the brain, which are things like the bones of your skull and the meningeal system. And then you also have biochemical protection or biochemical isolation by what's called the blood-brain barrier, which selectively isolates contents of the blood from crossing over into the tissue of the brain in the spinal cord, but mostly at the brain. So so that's what we want to look at first. And so the first thing, of course, is the bones of the cranium, and I think that sort of goes without saying, right? Your skull is this nice hard structure that's going to provide protection, outer layer of protection to the delicate nervous tissue beneath. Okay. After that, then you also have what we call the cranial meninges. Okay, cranial meninges, same thing we saw at the spinal meninges. It's just extended up into the brain. So same thing, has those same three layers: dura, arachnoid, and pia mater. And again, are going to help protect the brain from cranial trauma, as this dura mater not only wraps and isolates the brain, but it also sort of anchors it inside the skull, so it's not like bouncing around and moving around as much. And you can see in this image, you have the skull here, and then you have a lot of space, right? That's where you have those different layers of meningeal layers, but then also you have some CSF and other structures as the cortex, the brain is already there. So you have this nice space there to not only cushion and protect it, but also prevent injury from occurring. All right. So another way the brain is protected and supported is the CSF. And the CSF, as I said, is found in these ventricles, but it also surrounds all exposed surfaces in the CNS. So it's going to help cushion and delicate these structures, going to support the brain. Also a route for nutrients and chemical messengers and waste products. So the CSF playing a very important role. But again, it's continuous with what we see at the spinal cord. So you can see why doing a spinal tap, drawing off some CSF down at the lumbar region of your vertebral column can be beneficial or give you clues to what's going on up in the brain, right? Because the CSF is all continuous throughout the system, all right? All right, so CSF comes from what's called the choroid plexus, which is a combination of ependymal cells and capillaries. It produces and secretes this CSF into the ventricles and also remove waste products. And so this choroid plexus, you'll have an opportunity to see this when we do your brain dissection, sheep brain dissection coming up. You'll be able to actually tease some of that out of the ventricles. But that's where it's coming from. The CSF is coming from this choroid plexus, and it's found in these ventricles. So the CSF circulates from the choroid plexus, where it's originated, through these ventricles, down to the central canal of the spinal cord, and then into these apertures that allow it to go around the brain and around the spinal cord and cauda equina as well. So again, not only is it inside those ventricles and central canal of the spinal cord, but then it goes outside that location as well. And so this image gives you a nice overview of the flow, right? You can see the choroid plexus in these locations producing the CSF. It's circulating, obviously, down into the central canal of the spinal cord, but also through these apertures, it's going to move around the tissue as well, which is why you can get it all the way down here, right? Lumbar puncture into the subarachnoid space is going to allow you to grab some of that CSF off and give you a feel for what's going on inside the brain. Okay? Here is the dural different layers of the meningeal system and the circulation through those arachnoid granulations and the dura mater. Okay? You can see those locations there. All right, so another really important component with protection in the brain is blood supply. Blood supply is essential, of course, to the brain. This is how the brain tissue gets nutrients, like sugar is the primary nutrient that the brain relies upon, but also oxygen. Right? Without oxygen, you're not going to be surviving very long, or that nervous tissue is not going to be surviving very long. So really essential, the blood supply to the brain. It's delivered by the internal carotid arteries and vertebral arteries, and are removed by the internal jugular veins. And these images, you don't need to memorize these. You're going to be doing this in AP2 when you learn all these vessels. But it gives you an idea of the arterial blood supply via the common carotid, how it goes up into the brain, and what we call the cerebral arterial circle, or the circle of Willis, and delivers all this oxygenated blood and nutrients to the neural tissue, and then is drained via the internal jugular back to the heart, where the whole process can start over again. So those are just images to help you see the blood supply coming to the brain, which is obviously very significant and important. Cerebrovascular disease, then, is some sort of interference of blood circulation to the brain. And so a stroke or cerebrovascular accident is when you have a portion of blood to the brain that has been blocked or shut off, right? And so as a result, the neurons are not going to survive, and you get an area of neuronal death in that location. And they can range, right? You can have a hemorrhagic stroke, which is bleeding of, rupturing of the vessel, which could cause widespread damage, or you can even have what they call a TIA, trans-ischemic attack, which is a much smaller, or sometimes they refer to as mini stroke, where the impacts may be very temporary and happen temporarily, but then go away as the time goes on. So there's a big range that can happen with these strokes. But again, lack of blood flow means no oxygen. No oxygen means neuronal death. All right, so we've established that this blood flow is very important, obviously, for the nutritional and oxygen components, but also for waste products, etc. So this blood supply is really important. However, the content of our blood has so many things, from different proteins and other components and hormones and all types of things in our blood, that what's in our general circulation typically cannot just cross over into the central nervous system. Okay? And so it needs to isolate or be selective of what's going to be allowed to cross over into the nervous tissue. So we collectively call that the blood-brain barrier, where the central nervous tissue is isolated from the general circulation. This is formed by a network of tight junctions that basically it's very selective, essentially. It only allows certain compounds, like lipid-soluble compounds, like oxygen and CO2, of course, certain steroids, certain prostaglandins, to diffuse into the brain and the spinal cord, right? Everything else is going to be kept out. And so we call that the blood-brain barrier, because if you allow these things that are in the general circulation, it would be far too toxic and potentially damaging to this nervous tissue. And so astrocytes, if you recall, that was a type of glial cell, helps establish and control this blood-brain barrier by releasing chemicals that regulate that permeability, meaning only certain things can cross over into the nervous tissue from the general circulation. Same thing here with blood CSF barrier, right? Because you have this, remember, the choroid plexus, you have capillaries and the ependymal cells making up the choroid plexus. And again, it just allows the chemical composition, what you see in the blood versus the CSF, to differ. So what's in your CSF is not going to be exactly like what you're seeing in the blood, even though there is this sort of exchange that goes on at those capillaries. Okay? So there are 4 breaks in the blood-brain barrier. Okay? You have, and most of this is due to hormones, right? As we said, there's sort of this neuroendocrine relationship established in the brain because you have endocrine organs in the brain. And so, like, for instance, the pineal gland is a endocrine organ that releases melatonin, which is a pineal secretion. Okay, you need a break there. The pituitary gland is also attached to the brain. Okay, and so hormones like ADH and oxytocin need to be released, and then portions of the hypothalamus to allow for hypothalamic hormones, and then again the choroid plexus, which we just talked about that. Okay, so those are your breaks in the blood-brain barrier or blood CSF barrier where that's going to occur. All right, so to summarize, the meninges are going to help stabilize the brain within the skull. CSF is obviously providing a lot of functionality, but also, since it's completely surrounding the nervous tissue, can help prevent the brain from moving around, any sort of sudden movement. CSF, but as I said, also provides nutrients and removes waste products. And the blood-brain barrier and blood CSF barrier, again, selectively isolate the brain from things or chemicals in the blood that might impact the nervous tissue or disrupt the neural function. All right. So now that we have all of that complete, we can now start to take a look at all the different components of the brain. Okay, and we can start by looking at, since we just finished the spinal cord last chapter, well, now we can start with spinal cord ends, and that's the medulla oblongata. Go from the medulla up to the pons and the cerebellum, mesencephalon, diencephalon, and up to the cerebrum. So we want to just basically find out what these are all about, how they're organized, etc. So as you can see, the spinal cord ends and the brain begins with the medulla oblongata. And as you can see, there are 4 cranial nerves that emanate off of the medulla oblongata. Cranial nerves 9, 10, 11, and 12. You'll be learning about these cranial nerves, as I said, coming up in the next lab units. But you can see 4 of them are coming right off of the pons, or excuse me, the medulla. 4 of them are also coming off of the pons. All right, so the medulla oblongata, the first part we want to look at. This is the site, as I said, of spinal cord connection with the brain. It's a relay station, so it's going to do the nuclei of the medulla are going to be dedicated to relaying sensory information up to the thalamus, which we'll talk about the thalamus a little bit later. Olives are going to relay sensory information over to the cerebellum, but also can house the nuclei for those cranial nerves that I mentioned, but also regulate reflex centers like cardiac, vasomotor, respiratory, digestive, things like that are going to be controlled through the medulla. So medulla has some functionality, again, not quite as complex as we're going to see with other parts, but that gives you an idea of some of the function of what we're going to see with the medulla. Okay, and so here you can see the olives, right? Here you can see the pyramids, and those are going to regulate, or transmit or relay sensory information to the different parts of the body. Next part on top of that is the pons. Okay, the pons is going to link the cerebellum with the rest of the brain and spinal cord. Has 4 cranial nerves as well, 5, 6, 7, and 8. And so through those can help regulate eye control, respiratory center, sleep center in terms of its function. But the biggest thing is that it sits directly across from the cerebellum, so it provides sort of a functional and structural link for the cerebellum with the rest of the brain and the spinal cord. So that's another big function of what we're seeing with the pons. So the pons sits right on top of the medulla, and here you can see, right, here is the cerebellum, here is the pons, and you can see that relationship there. You can also see the 4th ventricle in that location. All right, so there's the pons right there. All right, so on to the next part of the brain, and that is, since we're at the pons, we'll go directly across from that, and that is the cerebellum, right? Cerebellum is an automatic processing center that helps with movement, essentially, okay? The origin of our movements is not based in the cerebellum. Those come from the primary motor cortex up in the cerebrum. But the cerebellum plays a really important role in adjusting and programming and fine-tuning these movements so that we can maintain posture and balance and make our movements very smooth and coordinated and organized. That's coming from the cerebellum. Okay? So really important functionality with the cerebellum in terms of regulating and fine-tuning those movements. So some structures of the cerebellum: the folio, which is the surface of the cerebellum containing highly folded neural cortex. The vermis is sort of a slender band of cortex that basically divides the cerebellum into 2 hemispheres. So kind of like we'll see at the cerebrum. The cerebellum has these left and right hemispheres, and if you sort of take a sagittal section of the cerebellum, you're going to find what appears to be look like a tree, essentially, tree branching. But that's just the white matter branching inside the cerebellum, and we call that the arbor vitae, or tree of life. And you can see that arbor vitae right there. Okay? And again, you can also see the relationship between the pons and the cerebellum. Here is the vermis, which divides the 2 hemispheres, right? You got a right and left hemisphere of the cerebellum, and the vermis divides them. All right. So to really, sometimes it's effective to study what happens when an organ is dysfunctional to understand really what it does. And this is a great example of that. When you have a disorder of the cerebellum, when you have some issue with the cerebellum, you're going to see ataxia. Ataxia is a gross lack of coordination in muscle movement. Think about someone that's been intoxicated, or that is intoxicated, right? And unfortunately, we've all probably seen these videos or experienced it ourselves, perhaps, when we're intoxicated. You have temporary impairment of the cerebellum, and so what happens? Your movements aren't very smooth, you have poor balance, you can't coordinate your movements very well, and so muscle coordination is disturbed. But that's temporary, right? You could have more severe level of ataxia based on some sort of head trauma or stroke, where the cerebellum is impacted, but that's what you're going to see is ataxia. All right, so that's the cerebellum. So we've seen the medulla, the pons, and now the cerebellum, and now we can move on to the mesencephalon, or midbrain. As I said before, things are going to start to sort of increase in complexity as we move through this material, and that's where we'll start here with the midbrain, or mesencephalon. Mesencephalon has several things we're going to look at here. Primarily, we're going to focus on what's called the corpora quadrigemina, which are two pairs of sensory nuclei. And then we also want to talk a little bit about the substantia nigra, which is a pigmented area of gray matter that releases dopamine, which helps regulate our skeletal muscle tone. Okay? And those will be things we'll focus on for the midbrain, or mesencephalon. And so here you can see the midbrain, or mesencephalon in this section here. You also have one of the areas we want to talk about, which is the corpora quadrigemina. And you have two superior caliculi, and you have these bulges, or two inferior caliculi. And we'll talk about their function in a second. Here's a nice cross-section through the midbrain, and you can see nicely here is the substantia nigra. You can see the cerebral aqueduct right here. This is where the superior caliculi would be, and here are the cerebral peduncles over there. Okay, so some important structural components that we're going to talk about that relate to their function. So the first thing are those sensory nuclei, collectively known as the corpora quadrigemina. These corpora quadrigemina process both visual and auditory sensations, and ultimately lead to an appropriate reflex response based on that visual or auditory sensation. So you have what's called the superior caliculi and the inferior caliculi. The superior caliculi respond to a visual stimulus of some kind, typically like a pretty intense visual stimulus, like a bright light, and control the reflex movements in response to that bright light of like the eyes, head, and neck, for instance. So think about what happens when, maybe you're in a dark room and someone turns on a light you weren't expecting, or you get a really bright flash of light in your eyes that you weren't expecting, right? You might turn your head or your neck a little bit, or you close your eyes in sort of a reflex response to that very bright light. That is, reflex is, routed through the superior caliculi of the corpora quadrigemina. Okay, so it's a visual stimulus that's causing a reflex response to our eyes, head, and neck. Similarly, the inferior caliculi of the corpora quadrigemina do the similar type of thing, except with an auditory stimulus, like a very loud noise. Or think about like a really loud clap of thunder that you weren't expecting, right? What would be the reflex movement of your head and neck in response to that, right? You might duck your head down or shrug your shoulders up or something like that as just sort of a reflex response to that really strong stimulus, okay? And so that is the inferior caliculi of the, midbrain, or mesencephalon. Now, that's more at the roof of the mesencephalon. The lower portion of the mesencephalon, the substantia nigra, is something I want to talk about. And the substantia nigra, again, release neurotransmitter dopamine. And dopamine from the substantia nigra help inhibit what are known as the basal nuclei. This should sound familiar to you, because the basal nuclei were areas of gray matter that are deeper in the brain. Cerebral cortex or neural cortex on the surface of the brain, basal nuclei are deeper. And the basal nuclei are regulated or monitored or inhibited by the dopamine coming from the substantia nigra. And one of the functions of the basal nuclei is to help with our resting muscle tone, right? Stimulate those muscles. You have a certain amount of muscle tone, so you're not like a sack of potatoes just falling over, right? You're able to hold your head up and hold your body posture and those types of things. And so a lot of that subconscious regulation of skeletal muscle tone for that posture is coming from the basal nuclei. Now, if the basal nuclei aren't regulated by dopamine from the substantia nigra, those basal nuclei are going to become overactive or more active. And what you're going to see is an increase in muscle tone, right? Because that's what the basal nuclei do in this case, are give us our muscle tone. But if they're not regulated, right, that muscle tone might be too strong. And so as a result, you see this increase in muscle tone. That's what Parkinson's disease is, okay? This increase in muscle tone. And Parkinson's is a movement disorder, so this increase in muscle tone can lead to movement issues like tremor and shuffling and other types of issues in terms of as it relates to motion, generating motion, okay? And so that's what happens in Parkinson's disease. Again, the basal nuclei are responsible for subconscious control of muscle tone, also helping with the coordination of learned movements, and are regulated by that dopamine from the substantia nigra. If the substantia nigra secretes less dopamine, the basal nuclei become more active, and that's where you see an increase in muscle tone, which causes these movement issues. Okay? Here's a nice image sort of showing what it looks like, right? Here, this looks familiar to what we looked at before, right? You can see the substantia nigra really well in a normal, healthy patient, but in Parkinson's disease, right, there's not as much substantia nigra. And here's some actual tissue, right? Look at this one. There is no, substantia nigra is supposed to be right here. You don't see anything, right? But look at down in normal, healthy brain, right? You got a nice area, pigmented section of the mesencephalon. That's going to release dopamine, and that's going to help regulate the basal nuclei. Now, some people with Parkinson's disease display what's called a kinesia paradox, which that means that they can often do other types of movement activities without incident, right? Like singing, running, dancing, things like that, where typically just walking is a very difficult challenge. These types of things can be done without incident. So oftentimes people with Parkinson's are encouraged to do, a dance class or, ride a stationary bike or other types of exercise that they can do that typically they would not be able to do when they're, trying just to walk, for instance, okay? And the reason behind this is that perhaps another part of the brain sort of overrides this pathway, like maybe the cerebellum, which helps also regulate and coordinate movement and fluidity of motion. Perhaps that's overriding what's going on at the basal nuclei and allowing people to do these other types of activities. I'm not sure, we're still looking at it, but if you click on this link where it says PD patient riding bike, it shows a nice video of patients with Parkinson's disease, pretty severe Parkinson's disease, having a very hard time walking, and he's relegated to a wheelchair, unfortunately, but then tells his doctor that he can ride his bike, no problem, and the doctor is kind of amazed by this and asks him to show him, so they take a video of him riding his bike without incident. So it sort of presents this kinesia paradox. All right, so that's Parkinson's disease regulated by the substantia, or that's impacted when you lose the substantia nigra from the mesencephalon or midbrain. All right, so that's it for the mesencephalon. On to the diencephalon. Diencephalon sits on top of the brain stem and is the structural and functional link between the cerebral hemispheres and the brainstem and on down to the spinal cord. Okay, helps integrate sensory and motor commands. Has three regions: epi, thalamus, and hypothalamus. And so here you can see the general location of where we're going to have the diencephalon. Right here is the midbrain. Right here you can see the corpora quadrigemina. Over here there's the cerebral aqueduct. Substantia nigra are going to be in this region here. Here is the pons. You can see there's the cerebellum, so you get your bearings, so to speak. And the diencephalon has three parts. So there's an epithalamus, which is up here. The main central mass is the thalamus, and then down here, the lower part is the hypothalamus. And the hypothalamus is attached to the pituitary gland, which would basically be right here, essentially, as it's attached to what's called the infundibulum. So that links the hypothalamus with the pituitary gland, which is basically right there. All right? And again, to sort of tie what you're learning in lab, the pituitary gland, also known as the hypophysis, resides in the hypophyseal fossa of the cella turcica, which is the landmarks on the sphenoid bone that you're learning in lab. All right, so the epithalamus is the roof or top portion of the diencephalon. And the only significant thing that I want you to know is that this is where we see the pineal gland. Pineal gland is an endocrine gland that secretes hormones. That hormone is melatonin, which can help with regulation of day-night cycles, also contributes some to reproductive function. Okay, so that's the epithalamus. And again, the pineal gland will be another thing you'll be able to see when you do your brain dissection. The thalamus is the main central mass of the diencephalon. And the thalamus is really interesting because it is sort of a filter or sorting station of ascending sensory information going up to the brain. Okay, and so it relays this information to the appropriate location, and allows for the message to be sent. So it's sort of like a sensory filter or sorting station, right? It's sort of the last location before it gets up to the cerebrum. So information coming to the brain has to go through the thalamus before it's sent to the appropriate location. And so the thalamus is made up of thalamic nuclei, which are different types of thalamic nuclei. You have an anterior, medial, ventral, posterior, and lateral group. And you can see that on these images. And this is a really nice image because it sort of color codes what these little areas are responsible for. So, for instance, here you can see general sensory input coming up into this ventral group, right, in purple. And so that is then going to go up to where you have primary sensory motor cortex, which is that sort of dark purple area, so it matches, right? Visual input. You may or may not know that all of your visual stuff is processed in the occipital lobe. So the information coming from your eye via the optic nerve is going to the lateral geniculate nucleus first, and then it's going to go and be routed up to the occipital lobe. So it's sort of like a sorting station, right, to get information to the appropriate location. So this is sort of a close-up of those different regions. And this is a nice chart that sort of shows you, the function of these different nuclei and what their sort of target is, where they're sending information to. All right, and so some of the stuff we looked at. So that's a nice summary of the thalamus. And finally, you have the hypothalamus, which is the bottom portion of the diencephalon. As I said, this is a regulatory center, controls a lot of what's going on inside the body, and it's also attached to the pituitary gland via what's called the infundibulum. Okay, so the hypothalamus has what are called mammillary bodies, which help process olfaction and other types of sensory information. It has an infundibulum, which again is what attaches to the pituitary gland, and also has this tuberal area, which helps control the pituitary gland in terms of its function. Okay? So here's a nice shot. Here is the hypothalamus, here is the infundibulum, and here is the pituitary gland. Okay? So what does the hypothalamus do? Well, it does a lot. A lot of it is done through the pituitary gland, but a lot of this is regulated through the hypothalamus itself as well. So hypothalamus can provide subconscious control of skeletal muscle. Okay? We'll see regulation of some autonomic function, helps establish this relationship and activities between the nervous and endocrine system, so this neuroendocrine relationship we were talking about, because again, the hypothalamus, which is part of the brain, is directly attached to and communicates regularly with the pituitary gland, which is an endocrine organ. So really important in terms of coordinating between nervous and the endocrine function. It secretes hormones. There are two specific hormones come directly from the hypothalamus. That's ADH, or antidiuretic hormone, and oxytocin. We'll talk about those later on when we do the endocrine system. Can contribute to emotional and behavioral drives via the feeding center and the thirst center, coordinate autonomic function. I already sort of mentioned that one, and can help regulate body temperature. Right? We talked about body temperature regulation back in AMP1. We looked at earlier in AMP1. We looked at thermal regulation, and so we said the hypothalamus is sort of that control center. All right. So that's it for the hypothalamus. And so just make sure you have a good understanding of the hypothalamus and all it does, and all of its functions. Before we move on to the cerebrum, I briefly want to mention what's called the limbic system. The limbic system is a functional system rather than an anatomical system. A functional system means it draws on different regions of the brain. It's not just one area that's responsible for something. So the limbic system, also known as the motivational system or your drive or desire, comes from these different regions, part of the cerebrum, part of the diencephalon, part of the mesencephalon. Helps establish emotional states, maybe facilitate memory storage, that gives us our motivation and our drive and desire to do things. All right, so that's really all you need to know about the limbic system at this time. I just want to briefly mention that before we move on to the cerebrum, which again, the cerebrum is the largest part of the brain, right? This is where all the higher order stuff happening, all conscious thoughts, intellectual functioning, processing sensory and motor information. So we're going to see a lot of functionality with the cerebrum, of course. And so, as I said, the gray matter, there are two areas. You have cerebral cortex or neural cortex, which is on the surface of the brain. Then you have the basal nuclei, which are much deeper. Okay? The white matter, then, is pretty much everywhere else if you look at a cross-section of the brain. If you look at the surface of the brain, you're going to see these gyri, which help increase the surface area. They're basically folds. And the cells here just a shell, a depression that separate these gyri, and the fissures are just deeper grooves, like a longitudinal fissure divides the cerebral hemispheres, for instance. So, and then the central sulcus and lateral sulcus also play a significant role, which we'll talk about in a minute. But here you can see the gyri, right? Like this is a gyrus right here, and you can see how they're like folds, right? They're neural folds. And the space between them are the sulci, right? And so then you have a really important lateral sulcus here and a central sulcus here that help divide the frontal lobe from the temporal lobe and the frontal lobe from the parietal lobe. And we'll talk about that central sulcus a little bit more. But again, these regions of the brain, frontal, parietal, occipital, temporal lobe, coordinating with what we've learned in the skull, right? You have the frontal bone in the skull, parietal bone, temporal bone, etc., sit right on top of those locations. And of course, you can see the cerebellum, and the end of the medulla, and the spinal cord would basically be right there. And you can see part of the pons as well. All right, so this is a nice color-coded image of the brain, which sort of coordinates what we looked at previously when we looked at the thalamus, and you can see how it's sort of color-coordinated in these different regions. And so we'll be talking about this in a minute in terms of how it's organized. This is a nice sagittal section of the brain. Again, you can see this here is nice corpus callosum, which helps link the two hemispheres. You can see the thalamus here. You can see the mesencephalon, pons, cerebellum, etc., in terms of all those locations. All right, so the white matter of the cerebrum is really important because, as we said, a lot of what the axons do are communicate messages either within one hemisphere or maybe crossing over to different hemispheres. So there's a lot of sort of talking, if you will, amongst neurons just within the central nervous system, within the brain. And so we want to take a look at what these different types of white matter fibers, maybe these axons, how they can be classified. So classified into three different things. The first one is called an association fiber. And association fibers are fibers that connect different neurons and components within one hemisphere. So the curate fibers are very short that connect one gyrus to another, and longitudinal fasciculi are longer bundles that may connect, for instance, the frontal lobe to some other, occipital lobe in the same hemisphere. But they're not going to cross over. They're going to stay within one hemisphere. Okay? So that's association fibers. Commiseral fibers are another term for bands that are allowed to link the two hemispheres, right? So the corpus callosum I've already mentioned. That's our big, thick, white band that allows for communication to cross over and communicate between the two hemispheres. Anterior commissure is another location for that. And then finally, projection fibers are fibers that pass through the diencephalon that, again, help link the cortex with the diencephalon, but then the rest of the brain and the spinal cord as well. Okay? And so you can see that in these images. You see arcuate and longitudinal fasciculi. You can see the anterior commissure and corpus callosum. You can also see the longitudinal fissure here that divides the two hemispheres. And then you can also see projection fibers, right, coming from the diencephalon and projecting out to the appropriate region of the brain. All right, so the basal nuclei, another sort of component or feature of the brain, which also referred to as cerebral nuclei, were masses of gray matter that are deeper in the brain, and oftentimes they direct subconscious activities. We talked about the basal nuclei and its association with Parkinson's, right, and how they regulated skeletal muscle tone and sort of coordinated movements, things like that. So that's one activity of the basal nuclei. There are different regions of the basal nuclei. I'm not as concerned with that. I just generally want you to know that the basal nuclei are deeper, right? They're deeper into the brain. You can see on this image, right? The cerebral cortex is way up here on the surface, and the basal nuclei are these areas that are deeper. But you can also see the white matter that makes up the rest of the brain. All right, and so as I said, basal nuclei, one of the big things involved with is, again, muscle tone and coordination of limb movement. So you can see when these aren't monitored and regulated by the dopamine from the substantia nigra, you're going to have movement issues, right? All right. Onto the cerebral cortex, which is the top portion of the gray matter in the brain. And there are different regions that are responsible for different areas. And so two of the things we want to look at are what are the motor and sensory areas of the cortex. And motor areas are in the precentral gyrus of the frontal lobe, and it's also referred to as the primary motor cortex. Okay? Sensory areas of this region are in the postcentral gyrus of the parietal lobe, and also referred to as the primary sensory cortex. Okay? There are other special sensory cortices like the visual cortex, auditory cortex, olfactory, gustatory, right? Bringing all this sensory information, and they're processed in regions of the brain. And so this is a really nice image sort of showing that, right? The central sulcus, right? It divides the frontal lobe and the parietal lobes. In the frontal lobe, you have this dark area of red. That is the primary motor cortex, also known as the precentral gyrus, right? And so, if you want, your skeletal muscle to contract, for instance, you want to lift something, or you want to start walking, right? That impulse is going to originate in this primary motor cortex, work its way down through the spinal cord out to the muscle, and the muscle will contract. Same thing with the primary sensory cortex, which is in this postcentral gyrus, right? Information about general senses, right? Like if you touch something, you're trying to figure out what that is. That information will go from spinal, up your nerve to the spinal cord, up to the diencephalon, and processed up to the primary sensory cortex, and that's where that information is processed. Visual information, for instance, right? Things that comes from our eye and travels back, as I was mentioning, it goes to the occipital lobe, and so there's a visual cortex in the occipital lobe for that. All right? Now, to sort of increase the complexity of what information is processed in the brain, there also are what we call association areas. And sensory association areas monitor information arriving at the sensory areas of the cortex. Okay? And so, for instance, the somatic sensory association area is going to interpret input to that sensory cortex. So it can, for instance, recognize and respond to touch and understand what that is. There are visual association areas that can interpret what arrives at the visual cortex. Auditory association areas monitor and interpret what's arriving from our ears, the ability to hear. Okay? And so these types of association areas sort of increase the complexity of what the brain can do in terms of analyzing what's being sent to these areas of the brain. Okay? And then finally, integrative centers take it even a step further and take this information from the association areas and direct much more complex and analytical types of responses. So the general interpretive area, the speech center, and the prefrontal cortex are all examples of integrative centers. And just like I said, integrative, right? It's taking information from different parts and sort of making sense of all these things. So, first one is the general interpretive area, also referred to as Wernicke's area. This typically is only present in one hemisphere, and it receives information from all these sensory association areas and coordinates these responses and generally understands what's happening between what you're seeing and what you're hearing, and gives you this ability to sort of generally interpret what's being sent to your sensory area. The speech center is also referred to as Broca's area, is needed for, making speech that makes sense, right? Someone that, for instance, has a damaged speech center, like maybe had a stroke or something, it impacts the speech center, because it's typically only found in the left hemisphere. Maybe they can make sounds and actually have the ability to produce sounds, but they may not be able to make words or have the appropriate breathing and vocalization needed for normal speech, and therefore things won't be very clear, right? So really important area, but you can see it's sort of a higher complexity in terms of what it can do. And then finally, the prefrontal cortex integrates information from the sensory association areas and can perform abstract intellectual functions, things like predicting what may happen when you do something, which can lead to frustration, tension, anxiety, because of your ability to sort of infer what might happen based on what information is coming to your brain. So that's the prefrontal cortex of this frontal lobe. Okay? And so here we can see those locations in the brain: speech center, general interpretive area, etc. All right, last thing we want to talk about here is what we call hemispheric lateralization. And that means that there is actually functional differences between these left and right hemispheres. Basically, each cerebral hemisphere performs functions that may not be performed by the opposite hemisphere. And so just getting sort of a general snapshot, the left hemisphere is typically thought to be the dominant hemisphere. Most people are left-brain people, which controls reading, writing, math, decision-making, speech, language, all those types of things. That's why most people are right-handed, right? Because everything sort of crosses over to the other side of the body, okay? Right hemisphere is more sensory-focused, recognition, things like that. And so here you can see how this works, right? Like, so writing, for instance, is initiated on the left hemisphere, but as we said, things cross over to the other side of the body, and so the right hand would be typically in control of that. Things that you process in your left visual field are actually going to cross over and be processed in your right occipital lobe, things like that. And you can see the corpus callosum here, which allows for communication to cross over as needed. So just have a general understanding of what we're seeing by this hemispheric lateralization as things can cross over to different sides of the two hemispheres. All right, so this last part, don't worry about it. We're not going to do electrical activity of the brain. I think we have plenty to learn in this unit, so we're going to cut it there. Just finish up with that hemispheric lateralization. So that wraps the brain lecture, and then this will wraps this unit as well. And then in the last unit, we'll be focusing primarily on the autonomic nervous system, the senses of the eye and the ear, and then the endocrine system. All right? So that's it for now.